Next, we will talk about osmosis. So osmosis is a passive transport of water via specialized protein channels. They call again aquaporins, like in here, aqua, water, porins. And osmosis is type of the facilitated channel diffusion. Facilitated again because we need this is specialized protein in here. And movement of the water is spontaneous or passive occur from area of the high concentration of the water to the area of the low concentration of the water. So water moving to the area of the low concentration of the water. And osmosis, it's only about movements of water from area of the high concentration of the water to area of the low concentration of the water with help of specialized transmembrane proteins, aquaporins. And in here shows, so if we have in here outside of the cell, we have a lot of high concentration of the water, and inside of the cell, we have low concentration of the water. So water will move through these specialized transmembrane channels inside through aquaporins. Osmosis is a process by which molecules of water, which is solvent, tend to pass through the semi-permeable membrane from a less concentrated solution into the more concentrated solution until equalization balance will be achieved. So if we look right now on this example in here, we see left side and right side of the tubes. And this tube, how you can see, it's connected. So this is where they connected. This is, and what is this? This is will be membrane show. And this membrane is permeable. It means that both solute molecule and water can free penetrate. So water is solvent, and it's showing here just blue color in both sides, right and left. This is blue, this is water solvent. Red molecules in here, red molecules on both sides, right and left. So this is will be sugar molecule, and this is will be solute. So this is solute is a sugar molecule. And we will see that our membrane, permeable membrane in here, dividing this tube, and on different side, left and right, we have different concentration of the sugar in the solution. On the left side, we will see we have just few molecules of the sugar. So it means it's less concentrated solution on the left. And on the right in here, we see much more red molecules of the solute sugar, so it's more concentrated solution. And because our membrane, which connected these two sides of the tube, right and left, permeable, and both water can move back and forth, and our sugar molecules can move in both directions. So what will happening with time, we will see that equilibrium will happening. So we will see that on our left and right side, we will have exactly same concentration of the sugar. So now concentration of the sugar equal. We will see in here solute, this is our sugar molecule red, on the both side equal, and water on the both side equal, and we have exactly same level in here of the volume of this solution. So when we will have more concentrated, like in here in the red side first, more concentrated solution, so it's called that we have greater osmolarity in this right part. When we have on this left solution, we have just few molecules of the sugar. So it's a less concentrated solution of the sugar. So it means this solution have low osmolarity. So this is what is this talking about. So concentration of the sugar and osmolarity is correlated, how you can see. But it will be different situation if on our left and right side of the tube, if we put exactly same solutions in here, higher concentration, low concentration of the solute, this is red particles in here. But if our membrane in here, this membrane, if this will be selectively permeable or semi-permeable, the other name for this, and only water can penetrate and cross, only water can cross. So what will happen in this case? So over time, we will see that water from the left side will move into the right side, because we have higher concentration of the water on this side, on the left, for the water compared to the right side. On the right side, we have less concentration of the water. So this is why water will move to the right. And solutes, this is red, large particles, they will not be able to penetrate through this selectively permeable membrane. So in the end, what will happen that water leave to the right, and because of this, volume now change. So we have high volume on the right side compared to the low volume of solution on the left. But concentration or osmolarity of this solution on the left and right will be the same. Concentration of these solutes will be the same, and it means osmolarity of this solution on the right and left will be the same. So in here shows the same what we look on the previous slide. So this is selectively permeable membrane, and because of this only water was moving to the right, and this is equilibrium achieved. Now we will see why water moving in here. So if you look on this diagram below, so this is what explaining. So again, this membrane in here, these gray dots, this is selectively permeable membrane. Only water can penetrate. And on this left side, we have left and right. And in here, we have right and left. So on the left, we have low concentration of this solute, these sugar molecules, low concentration. On the right, we have high concentration. High concentration of this red and low concentration of this red. Yes, we have only one when we magnify this. On the right, we have three, so higher concentration. So this solution on the right will be hypertonic. Hyper means high concentration of this red solute. On the left, we have hypotonic, low concentration of this red. And water molecules in here visible actually, like separate. And you will see that water molecules, they are attached to these sugar, for example, molecules, red, on the both side. But you can see that on the left side, in the hypertonic solution, we have much more concentration of the free water in here, which is, it's called free because it's not attached to these red large molecules. It's not enough of them. On the right side, in the hypertonic solution, we have more, this is red molecules, and because of this, we have less free water. So in this case, we have in this On the left side, we have a lot of free water, and on the right, we have less of this free water. So we have on the left side high concentration of the water, and on the right side we have low concentration of the free water, when we're talking about only free water which is not attached. And because of this semi-permeable membrane, water molecules, we have a lot of these free molecules in here on the left, so they will move because of this to the right. And this will be continuous until concentration of the free water will be the same on the right and left side. And this is what's happening in here. Now concentration of the water in here is the same on the left and right side. So in this moving cartoon, you can see that we can see on the different side, on the left and right side, we have different concentration of these green large molecules, sugar molecules, and water molecules will move from one side on the other side. So this shows these movements of the water, osmosis, with selectively permeable membrane, from the area of high concentration of the free water to the area of the low concentration of the free water until equilibrium will be achieved. So in this diagram shown now without these cartoon movements, but you will see the same happening. We see semi-permeable membrane, only water can penetrate through this membrane. We have high concentration of the solutes in here, red, and we have low concentration of the solutes in here, and water molecules will move to the right. How they say this is red is a sugar, but you can say this is a salt, doesn't matter. But yeah, again, when osmosis happening, it's always follows this rule. Water follows the salt. So you always know if we have more, this is some kind of molecules other than water. Water will move to the area where is high concentration of these solutes. And this is what will happening. Its volume will change in the end because only water can penetrate through this membrane and solutes cannot penetrate. The same shown in here. So this is just phospholipid bilayer. This is the membrane in here show. This is pink. This is our aquaporins channels, and water can move freely, passively. And we will see in here, green is the sugar molecules in here. On the right, we have high concentration of the sugar. On the left, we have low concentration of the sugar. And water will move to the area where is high concentration of the sugar, or water, free water will move to the area where is low concentration of the free water. So next we will talk about effects of osmosis on the cell. And we will see that we have three different types of the solution. We have isotonic, hypertonic, and hypotonic solution. And in here there is division happening. And we have different cells. So as an example of the animal cells, in here we will see red blood cells, and for the example of the plant cell, we have one in here, cell, green cell. And what is special about the plant cell, that they have cellular membrane around the cell. which have, let's put it in the blue, aquaporins inside them. So this is me talking about green cells, yes, in the plants. But plants also have very strong and rigid cell wall. And this is what I'm making green in here. So this is the cell wall, and here it's a green, and in here it's a green. And cell wall lack aquaporins. Animal plants, on the other hand, in here, animal plants have only cell membrane, and they cell membrane have aquaporins inside of them for transport of the water. So now we will see what happens if we put animal cell, red blood cell, like in here, and plant cell in here, in the isotonic solution. So iso means the same. So it means same concentration will be of the salt, for example, inside of the cell and outside of the cell. So we have exactly same concentration. So it means water will move in and out of the cell freely. And it will be same amount of the water moving outside of the cell and same amount of the water move inside of the cell. Because concentration of the salt, for example, will be the same on the both sides. How I was talking on previous lecture, for example, 0.9% inside of the cell of the salt and 0.9% of the salt, concentration of the salt outside of the cell. So this is the same, isotonic. So water will be moving in the same amount in and out of the cell. So how we can see that size of these animal cells will be the same, no change in the size will be. But if we're talking about plant cell, so again, water in the plant cells located in the special organelle called central vacuole in here. And when we will put this plant cell inside of the isotonic solution, so concentration in this vacuole will be 0.9% of the salt, and concentration of the salt outside of the plant cell will be the same, 0.9% of the salt, because it's isotonic solution. So water will move in and out of this is central vacuole in the same amount. But if we look, and not only it will be moving after vacuole, it will moving also from the passing, yes. But if we talking about if we put animal cells in the hypertonic, hypertonic, it means that outside will be, for example, 9% of the salt, and inside will be 0.9% of the salt. So water will follow the salt, and we will have more water in 0.9%. So water will leave animal cells, and animal cells like this red blood cell will shrink. If we have in here plant cell, and we have the same hypertonic solution, so inside we have 0.9%, outside we have 9% of the saline salty solution. So water, we have more water inside than outside. Water will follow the salt. So water will move away from the cell to the area of the high concentration of the salt. water follows the salt. And you can see that central vacuole will change shape. It's become smaller because water is leaving. But if we compare the size of the cell in here, this is the one cell. Will change or not? No. Size of the cell, plant cells will not change. And why it is? Because we have very rigid cell wall. Cell wall, this is green, and it's not changing size. Only inside of the cell vacuole will change size. If we're looking on the next example, hypotonic solution, so we have low concentration of the salt in the solution. For example, it will be 0.1% in the solution, and in the cell will be 0.9% of the salt. So in this case, water will follow the salt moving inside of the cell. So size of the animal cells will change. Yes, it will increase size. It will increase size. It can even burst and be damaged. But if we put animal cell inside of the hypotonic solution, so what we will see? Water will again follow the salt. So water moving inside of the cell, inside of the central vacuole, and size of the central vacuole increased dramatically, very large. But if we look on the size of the plant cells, it's changing? No, it's not changing. The size of the plant cell in any solution the same. Why? Because plant cells surrounded by this is very sturdy cell wall, which is not allowed to change size of the cell. Okay, next we will see how you can distinguish what's happening in these beakers, osmosis or diffusion. So if we look on the beaker number 1, so beakers, this is a beaker, like big glass, and we see we have in here separation. So this is a membrane in here. And on the both side of the beaker, we have solution. It's called isotonic solution. It means concentration the same. It means water will pass through this membrane in the both concentration in the same amount, and its volume will not change. We have water, it's moving passively, same from one side, from the left to the right, in the same amount. Nothing changing. So this is diffusion of the water. Water just distributes themselves equally on the both sides of the semi-permeable membrane in here. But if we put in the second beaker in here, we have again semi-permeable membrane. And we have first water, same isotonic solution, but then we will put urea molecules in here, big, large, this is green molecule, on the right side. And now what's happening? If we put these molecules, they cannot penetrate semi-permeable membrane. And water molecules on the right side will be attached to the urea. And here they're attaching to the urea. They become not free anymore. So what happened now? Concentration of the water on the both sides is different. We have more free water molecule on the left compared on the free molecules on the right side of the beaker. And because this membrane semi-permeable, urea cannot move to the left side, but water can, and it will move. And when this is happening, what's showing in here on the last beaker number 3 in here, these movements of the water to the area where is less concentration of the water from area of the high concentration of the free water to the area of the low concentration of the free water. This is osmosis, because we have, in this case, in the beakers, different concentration of the urea, and it's make water to move, and osmosis happening. So this is how you can distinguish. So diffusion, it's in here, show passively. Osmosis also passively, but it's to normalize. This is concentration of the urea, which cannot penetrate through the semi-permeable membrane. So on the beaker number 2 and 3, this is showing osmosis. Again, when we're comparing osmosis and diffusion, you can say that when you go to the bakery, for example, and you smell in here concentration of this is fresh bread. Yes, so in here show these molecules high concentrated, and they will move from one area passively to the other area, whereas it's low concentration. So this is what's happening. This is diffusion. Osmosis, it's always about water molecules, and we need semi-permeable membrane in order to demonstrate it and show this, how it will move. And it's moved from the area of the high concentration of the free water to the area of the low concentration of the free water. And this is what osmosis will be. The other example of the diffusion and osmosis. So if you have hot water and you're making hot tea, or in here on the other you have ice water and making iced tea. So what's happening during these processes of the making tea with different temperatures also, it's diffusion happening in here. So, and again, diffusion depends on the temperature, how I was talking previously about. So, mechanism of osmosis occur when the medium surrounding the cells have higher water concentration than the cells. The cell will gain water along with all other important molecules and particles which dissolve in the water, and it's important for the growth of the cells. It's also occur when water particles move from one cell to another cell. So what is the significant importance of this? In animal, osmosis influences the distribution of the nutrients and release of the metabolic waste product. In plants, osmosis is partially responsible for absorption of soil water and for the elevation of this water to the leaves of the plant. In here show, so this is hair cells in the roots, and water will be absorbed through the osmosis inside of this root. So in here show root hair, and the water will be absorbed and then distributed between the cell, from one cell to the other, and passing like that. So water always moves to area of the low water concentration, because soil, if we put a lot of water in the soil, so water will be absorbed by the plants in here. Root hair will take in water from the soil by osmosis and distribute it even between themselves also. So in here show how water together with nutrients move by osmosis from the soil to the roots, and then distributed between cells and moving to the leaves, where is evaporation happening. So overview of the diffusion. Diffusion is movement of the molecules across membrane. And there are three types of the diffusion: simple, facilitated, and osmosis. Simple diffusion, when particles move from area of the high concentration to area of the low concentration along the concentration gradient. Example, oxygen, carbon dioxide gases. Facilitated or channel diffusion involves the protein channels which letting molecules through the membrane. An example for this will be potassium ions. In facilitated diffusion, a protein carrier changes its conformation to let passing molecules through, for example, glucose. Osmosis is a passive transport of water via specialized protein channels, and it's type of the facilitated channel diffusion.